Hi, I'm Alexis Rimbaud, Head of Research and Product Development at Random Walk Imaging. I'm going to talk to you about diffusion tensor distribution imaging, or DTD, one of the methods implemented in our data analysis software, DViewer. Let's start with a quick refresher on how diffusion relates to tissue microstructure. In this example, a drop of ink has been released in various diffusion media. The first diffusion medium features no obstacle whatsoever and is completely free diffusing. The second one features isotropic cells that are densely packed, and the last one features elongated cells. And you can see that after a few hundred milliseconds, the diffusion pattern of the ink drop will feature a certain size and a certain shape. The size of the diffusion pattern will depend on the average cell density surrounding the drop, and the shape of the diffusion pattern will depend on the shape of the underlying cells. In the brain, these environments could be like white matter, gray matter in a simplified way, and cerebral spinal fluid. To better characterize the size and shape of diffusion patterns, let's introduce this size axis, which is related to the inverse cell density, and this shape axis, which is related to cell elongation. When shape is not spherical, orientation becomes a relevant degree of freedom. To quantify these different properties, you can use, for example, the parallel diffusivity or the perpendicular diffusivity. You could also use the isotropic diffusivity, which is related to the trace of the diffusion tensor, or you could use this d delta number, which is a unitless number that goes between minus 0.5 for planar tensors, to 0 for spherical tensors, to 1 for stick-like tensors. One of the main problems faced by diffusion MRI is subvoxel heterogeneity. Most diffusion MRI voxels contain collections of distinct diffusion patterns or diffusion tensors. For example, if I take this coronal slice of the brain and I isolate one voxel in there, this voxel may encompass, for example, cerebrospinal fluid and also fiber populations. While the size of typical diffusion MRI voxels is of the order of millimeters, the size of typical biological structures is of the order of micrometers, which implies that in any given voxel, you will find a very diverse collection of diffusion patterns. The main question then is, what are the features of such collections? Let's talk about the statistical descriptors of the diffusion tensor distribution. These are different voxel contents which can all be characterized by a distribution of diffusion tensors, or a distribution of these isotropic diffusivities, normalized anisotropies, and orientation theta and phi. These various voxel contents can be characterized by three main features, and these features will be quantified using statistical descriptors. Their first feature is the diffusivity one, that is linked to the size of the diffusion tensors or diffusion patterns. The second one is the anisotropy feature, which is linked to the shape of the diffusion tensors. And the final one is the orientational order, which is linked to the alignment of the diffusion tensors. For instance, these two voxel contents feature roughly the same size and the same shape, but not the same alignment. To better understand statistical descriptors, let's remap this voxel content in terms of this distribution of points. Here you have the log of the isotropic diffusivity, and here you have the log of the ratio between the parallel and the perpendicular diffusivities. This indicates anisotropy as being positive for prolate tensors, 0 for spherical tensors, and negative for oblate tensors. You see that these two populations are characterized by a pocket of components here at 0 anisotropy and pretty high isotropic diffusivity, which is this population, and a second pocket of components here that has high anisotropy, prolate anisotropy, and a lower isotropic diffusivity. And the color gives you orientation with the same color coding as here, so you see that mostly green orientation. The first type of statistical descriptors is, for example, the average of the isotropic diffusivity, or the average of the d delta squared, which is this normalized anisotropy. This is equal to the mean diffusivity, and this is linked to the microscopic anisotropy. A higher order of statistical descriptors consist of the variances of these quantities. So you have the variance of isotropic diffusivities, which is linked to the variance of cell densities, and here this variance of normalized anisotropies, which is linked to the variance of cell elongations. Finally, you can quantify the covariance between these two quantities to understand whether or not cells that have higher anisotropy also tend to have higher isotropic diffusivity or lower isotropic diffusivity, or if no correlation between these two quantities exists. And now the question is, how can one estimate the features of these distributions? One of the most advanced methods for estimating diffusion tensor distribution is called diffusion tensor distribution imaging, or DTD. Considering this voxel content, the signal arising from it can be written as S0 weighted by all of the contributions from all the diffusion tensors within. In this case, the integral is just a continuous sum of these diffusion tensors. You see here that the signal decay from each of these diffusion tensors for a given B tensor or B matrix that you acquired is weighted by the distribution of diffusion tensors P of D. Diffusion tensor distribution imaging simply discretizes this equation as such. The index M corresponds to the index of acquisition. For example, here you have the mth acquired B tensor or B matrix. The sum here is discretized over the indices n, and this index n is for each of the diffusion tensors that you find here, this dn. And wn is the probability weight associated with dn. This probability weight is technically normalized to S0, as the sum of the different wn should sum up to S0. If you divide by S0, it is actually a signal fraction or probability weight. This equation can be rewritten in a more compact way. Here you have the column vector of signals, the column vector of weights wn, and this is the kernel matrix whose matrix elements are given by this exponential decay. In this framework, the voxel content that is shown here translates in terms of this distribution of points that I showed you earlier. Keeping in mind this S equal KW relationship, I'm going to go through the different steps underlying the diffusion tensor distribution imaging algorithm. First, you have the proliferation step, which consists in generating a random set of n-in diffusion tensors. Then you can compute the associated vector of weights wn for this set dn using a non-negative least square fitting. Finally, you simply save the components with positive weights. This entire step is repeated np time, number of proliferations. Next step is called mutation. We perturb the previously saved components. Each mutated components are put in competition with the original components in this extinction phase. First, we compute the weight vector wn using non-negative least square fitting, and finally we save the components with largest weights. This step is repeated nm time, number of mutations. The very final step of the algorithm is to save a certain number n out of components with largest weights. This number n out is typically set to be pretty high, around 50, to make sure that it does not impact the retrieved distribution of diffusion tensors. Now let's see the final result output by the diffusion tensor distribution imaging algorithm. This voxel features three populations: a low diffusive isotropic one, a high diffusive isotropic one, and finally a population of sticks. The DTD algorithm will output a solution which greatly fits the acquired signals. In this example, the red dots are the acquired signals and the black dots are the signals predicted from the solution retrieved by the DTD algorithm. This solution can be presented in this way. Here you see that these very thin lined ellipses here, here, and here denote the actual ground truth solutions, and these thicker circles on top are the solutions retrieved by the DTD algorithm. The radii of these circles gives the probability weights, Wn. Of course, you notice that the DTD algorithm features a lot of randomness, which means that the solution that we find is not unique. And this is actually a key feature of the problem at hand. Laplace transforms, which is the kind of transform we are currently solving, do not feature unique solutions. This DTD algorithm actually acknowledges it and embraces it, and can be simply repeated in order to find different solutions that all fit the diffusion signal to the same or to comparable extents. A more refined solution of our problem can then be defined, for instance, as the average of these different solutions. And a sense of uncertainty can be gained by computing, for example, variances across these different solutions. DTD relies on a minimal set of assumptions. Actually, there's mainly one, that non-Gaussian diffusion effects from restriction and exchange are negligible. In particular, there are no compartmental constraints, and all cumulants of the signals are considered. DTD also requires minimal regularization. Although non-negative least squares fits promotes rather sparse solutions, no additional regularization term is to be found. The weights are also imposed to be positive numbers, hence the non-negative least squares fits. This is a key property of weights, that they have to be positive. Finally, only a finite number of components are kept at the end of the algorithm. That was the n-out parameter I mentioned previously, and as I said, this number is kept pretty high to avoid any inconvenience. Let's look at an application of the DTD algorithm in breast cancer. For this, I'd like to acknowledge our collaboration with the Memorial Sloan Kettering Cancer Center in New York, and in particular the work of Isaac Daniel Naranjo. This is a non-diffusion weighted signal map of breast seen from top. Without being able to see it, there's actually a breast tumor here. I isolated 3 different voxels, and with DTD you can actually access the voxel content in terms of these diffusion tensor distributions. For example, the content of this voxel consists of diffusion tensors that have a high isotropic diffusivity and a low anisotropy. On this y-axis, the anisotropy goes from 0 for spherical tensors to 1 for stick-like tensors. In other words, these diffusion tensors are roughly isotropic and quite big in size. In this other voxel, the main contribution comes from this population of highly anisotropic diffusion tensors, which feature a lower isotropic diffusivity compared to these diffusion tensors. These are stick-like diffusion tensors of fibrous tissues. In the final voxel, the main contribution comes from this population of roughly isotropic diffusion tensors that also feature a lower isotropic diffusivity compared to these diffusion tensors. While being comprehensive, this way of representing information is not practical. So we need to make sense of the distributions one way or another, at least a way that enables to create maps out of them. A simple way to do so is to consider the space of diffusion tensors and to subdivide this space into different bins. For example, this big bin consists of all components that have very high isotropic diffusivities. In the brain, this bin will encapsulate diffusion tensors linked to cerebrospinal fluid, for instance. The thin bin encapsulates any diffusion tensors that has a very high anisotropy. In the brain, that would be white matter. Finally, the thick bin corresponds to diffusion tensors that are not very anisotropic nor very diffusive. Such diffusion tensors could be associated to densely packed isotropic cells, like in gray matter, for instance, but they could also be associated with systems like edema or like necrosis that all feature very small anisotropy and also quite limited isotropic diffusivity. In the breast, a segmentation maps of these different bins looks like this. If you forget the bottom of this map, which for practicalities of breast imaging has quite a low signal-to-noise ratio, SNR, the healthy breast tissue here looks blue or cyan, which is a mix of blue and green. This tells us that diffusion is high isotropic diffusivity in the blue voxels, and is a mix of diffusion environments that have low and high isotropic mean diffusivities without having a very high anisotropy. The tumor is made of a shell of fibrous tissue in red, associated with a thin bin of anisotropic diffusion tensors, that surrounds a core of thick diffusion tensors, non-anisotropic nor diffusive diffusion tensors. This core is actually necrotic tissue. Beyond the S0 and the segmentation maps, other maps can be defined based on the distribution of diffusion tensors retrieved within each voxel. For example, here you have the average, the iso, and the delta square maps. So this is the mean diffusivity map, and this is the microscopic anisotropy map. You can see that the tumor features a very low mean diffusivity compared to the healthy breast tissue, and here you can retrieve this shell of fibrous tissue in the microscopic anisotropy. If you wonder what fractional anisotropy looks like, it looks like this. And you see that except for a very tiny part of the tumor, or actually the shell of the tumor, the rest of the tumor is completely invisible. This is linked to the fact that these anisotropic cells here don't have a coherent orientation over the voxel scale. They're randomly oriented. Other bin resolved maps can be defined. Here, for example, the intensity of these maps is given by the fraction of components falling into the big, thick, and thin bin. And the color of these maps is given from blue to red by the mean diffusivity of these components pre-selected in each bin. So you see, for example, the big bin, which is defined as the bin of very high diffusivity components, is in red. In particular, you can see also that the core of the tumor does not feature the same color as the thick part of the healthy breast tissue. This other map is similar to the previous ones, but the color is now given by this index of microscopic anisotropy from 0 to 1. And of course, the thin bin, which is the only bin made of very anisotropic tissues, is the one shining in yellow, while the other ones are blue. Finally, we can define these mean orientation maps, which is only relevant really for the thin components because these are anisotropic and then features a relevant orientation. And if I focus on the shell of fibrous tissues I identified earlier, you can see that just by happenstance, the top of the tumor here features a pretty coherent green color, while the rest is roughly white. And this indicates that these elongated cells are roughly aligned together, while these are randomly oriented, which also explains why the FA becomes dark in these areas. Note that the microscopic anisotropy is quite iso-intense in this shell. Other maps can be defined, for example, the variance of isotropic diffusivities, which is linked to the variance of cell densities, or the variance of d delta squared, which is the variance of cell elongations, and finally the covariance between isotropic diffusivity and d delta squared. These parameter maps can also be obtained in the brain, for example using our data analysis software Diffusion. In this case, the dataset consists of a brain featuring a grade 1 meningioma in the left temporal lobe, and it was acquired by United Imaging in China. The different contrasts here are the mean diffusivity, the microscopic anisotropy, the variance of isotropic diffusivities, which is linked to the variance of cell densities. You also find these bin resolved maps. This is the segmentation map, where the blue is for the big bin, high isotropic diffusivity, so cerebrospinal fluid. The red is the thin bin of high anisotropy diffusion tensors, so the white matter mainly. In the green, this is low isotropic diffusivity and low anisotropy in the thick bin, so mainly gray matter and the edema on which I'm going to come back. You see also here the average orientation map in the thin bin. So this is the average orientation computed only within the thin, very anisotropic components. And finally, this is the colored mean diffusivity map in the thick bin. And you can see here that in the thick bin you have this gray matter and you have this other area that lights up at higher color, this yellow compared to the blue of the gray matter, which means that this area has a higher isotropic diffusivity. This is actually edema. Thank you very much for your attention.